Good day to everybody. Today, my topic of discussion is human blood group. This is very important human blood group because during the surgery and any accident happen, when the excessive bleeding will be happen, then you have to transfuse the blood to the patient so that for the survival of the patient. So it is very important. So before transfusing any blood to the patient, we have to determine, we have to check the blood group primarily. It is most important part of the treatment. So actually, what is blood? Blood is a body fluid which transports nutrition and oxygen to all cells of the body and carries away the carbon dioxide and wastage material. So, and now we look at the what is the composition of blood. Blood generally, I am talking about the human blood. The human blood generally uh, there are two parts of human blood. One is the blood plasma, which is the 55% of the blood, and another is the blood cells, which is the 45% of the blood. This is the general composition of the blood, human blood. And here your WBC, white blood cell, among blood cells, white blood cells, red blood cells, RBC, and the platelets are present. Platelets are present. Now this blood plasma, in the blood plasma, about 55% of the blood plasma, it is 90% is water, 90% is water, and 10% is other solid and gaseous material. Solid and gaseous material. Now we know that in WBC, in the WBC generally we in a healthy man or woman, the blood quantity of the blood quantity is generally near about 5 liter. We are carrying 5 liters of blood, and this blood, the pH of blood is generally 7.3 to 7.4. PH of blood is generally 7.3 to 7.4, and the quantity of blood is 5 liters, near about 5 liters. Now, uh, and in among the blood cells, the RBC actually, RBC, the function of RBC, red blood cell, is to carry the oxygen to the all the parts of the body, and the main function of WBC is the to develop the immunoresponse. Any foreign pathogenic bacteria or any virus, when are attacking our body, when are invading our body, this WBC actually fights against them. And among WBC, there are so many granules are there, types of WBCs are there. They are neutrophil, eosinophil, basophil, lymphocyte, and monocyte, and all are helping in the immuno response of body. And platelets are generally helping in the blood clotting. So there are different function of these blood cells, and in blood cells are this blood are generally altogether blood, the color of the blood is, you know, that red. But this when we separate the blood cells from blood, the blood or plasma will be left. and the color of the blood plasma will be yellowish color. Yellowish color due to the presence of bilirubin, carotenoids, and other transferrin like pigments. So, and among this blood plasma, 90% is water. When we are talking about the generally RBC are more in number in blood, and the WBC and platelets are less in number. Now, when this WBC are actually fighting against the any germ enter in our body, foreign body, they are actually fighting against this pathogenic germs. So the WBC, when any we are invaded by some pathogenic bacteria or virus, then suddenly the WBC quantity, WBC number of WBC in blood will be increased. This is a very normal thing, and this thing is called leukocytosis. Leukocytosis, and when but beside this condition, when any infection is not there, any foreign invasion is not there, but your WBC will be increased. In this case, it is called leukemia. Leukemia means blood cancer. And when any due to the any disease or the viral infection, when the WBC count will be less in blood, generally this is called leukopenia. Leukopenia. So this is the some terminology are related to this blood. Now we are talking about the blood group. Our today's topic is blood group. This is the general idea of blood I am giving uh, before discussing about the blood group. It will be easy to understand. Now, before going to the blood group, one thing I have the basic thing of immunosystem. Our immunosystem I am discussing about. There are two. Generally, there are so many barriers in our body to fight against this external pathogenic germs which are trying to enter in our body, to fight against them, to resist them, to protect, to resist them to enter in our body, so that we can we are not ill by this invasion of these foreign materials. Now, first, generally our in body, the immune system is divided into two parts. One is two systems. One is the innate immunoresponse, and another is another is the acquired immunosystem. And among acquired immunosystem, there are two types of system is there. One is the humoral mediated system, where B cells are involved. B cells are involved, and another is the cell mediated immunoresponse, where T cells are T cells are involved. Now, the B cells, why these are called B cell? Because they are matured in bone marrow. That's why this is called B cells. And T cells, why this is matured? These T cells are matured in thymus cell. Now, B cells, where the B cells immunosystem is growing, means acquired immunosystem. This system is called humoral immunosystem. And in this humoral immunosystem, one of the main component of this humoral immunosystem is the immunoglobulin or antibody, which is present in the surface of the B cell. Actually, they are present in the blood plasma. Blood is the liquid part of the blood. They are present from, they are extracted from the red blood cell. They are actual sorry, red blood cell, B cells. Now, they what they are doing, this antibody, when our body is attacked by any external pathogens, pathogenic bacteria or virus or any foreign materials, actually they will attack this antigen, foreign materials. Now, one important thing is this immune system is that there are two steps of immune system. Very important steps of immune system is the one is the recognition of self antigen from non-self antigen, and another is the response. This is the our in our discussion. This part will be very important. You have to be careful about that. This is the recognition of self antigen from non-self antigen. Means non-self antigen means foreign antigens. Our body will all my body will only respond when there will be foreign antigens. Antigens are generally protein or carbohydrate in nature. So these are foreign materials. But in our body also there are so many antigens, but they are self antigens. Antibody will not react with this body's antigen, self antigen. They will only react with the non-self antigen which are coming from outside environment, foreign particles. Now, when there is a good system of that to recognize the self antigen from non-self antigen, but sometimes what happen, this these features may fail and rise to the situation called autoimmune disease. So this is the thing, and this is the basic thing. Our body, our antibody in our body, which is fighting against any germs, actually they have the ability to recognize the self antigen from non-self antigen, means foreign particles. This is the main idea of this blood group, determination of blood group of human. Now, the antigens are present on the surface of the red blood cells. Generally antigens, this whatever I have we are talking about, the human blood groups are ABO group, ABO phenotypic blood group. Generally we are having this group. And now the antigens, this AB antigens, O is not any antigen, AB antigens are generally present on the surface of the red blood cells, and the antibodies are generally present in the plasma of the cell. Depending on these factors, Carl Landsteiner in 1900, in 1900, Carl Landsteiner classified the human blood into four groups. These four groups are A, B, AB, and O. So he has classified this, and this ABO was new several associations have been made between these phenotypes of ABO group AB, and O blood type. Generally, what is observed that the gastric ulcer is very common in O type individual, O type blood group individual, and the stomach ulcer and the gastric cancer is very common in the A type of blood group of human beings, individuals. And another thing is the lower number of one factor, von Willebrand factor. This is a proteinous matter, and it is responsible, it is helping in the blood clotting. So these are generally lower in the O group, O type group of blood group of individual. Now, this is the four types of blood group, and the occurrence of the blood group in the Indian population are generally like that. That this is the blood group, and percentage of occurrence in Indian population, first one is the O positive. O positive is the 35% of the population are O positive. Then A positive. A positive is the 30% of the population, and O negative is the 13% of the population. A negative is the 8% of the population. B positive is the 8% of the population. B negative is the 2% of the population. AB positive is the 2% of the population, and the AB negative is the 1% of the population. Now, this is the most abundant blood present in the Indian population, 35% O positive, and AB negative. AB negative is the least, most rare blood group found in the Indian population. When we any blood group will be considered as a rare blood group when the one population, one blood group, AB, one any group blood group will be found in the from the random sampling of the 1,000 people. When one happened in 1,000 random sample of people, so in that case AB negative blood can be considered as a very rarest blood group in Indian population. Now, come to the point. Generally, these are the alleles. These ABO phenotypes are encoded by the same genetic locus. Means this is the allele AB, and AB are the A, B, and O are the same allele of the same gene. So this type of things means one baby can acquire the one of the these three alleles A, B, O from each parent, and developing the nine possible genotypes and four possible phenotypes. How this can be happened? I am showing it by some diagram. It is the ABO phenotype acquired from mother, and this is the three phenotype A three allele. This is a three alleles, three genes of mother, and ABO genotype found in the offspring, means baby, offspring. And here is the ABO phenotype acquired from father, and these are the A, B, O. These are the three alleles of the father, and these are the three alleles of the mother. Now what happened? A can be one genotype, it means A ultimately. AB, it means AB. AO means A. AB means B. BB means phenotype B. Genotype BB means phenotype B. And BO means B. And AO, again AO means A phenotype. BO means uh, B phenotype, and OO means O phenotype. So from these are all the these 9 types of genotypes can be produced in the offspring. And no, 9, although it is 9, but actually it is giving the 6. uh, Actually this is the same AAA, so it is giving the 6 genotypes in from 9 genotypes, and 4 phenotypes ABO blood group means ABO. Now, what happened in the genetics here? Actually, the antigen A present in the red blood cells, I have told that the antigen A is present in the red blood cell, and B antigen is present in the red blood cells of B group blood group. Now, how this A antigen and B antigen will be present, like that I am showing by a diagram. Like this is group A, this is group B, this is group AB, this is group O. Now this is the red blood cells looking like that, and this is the A antigen present here, like that. A antigen presents here like that, and the antibody will be B antibody. When A antigen will be present, the antibody present in the plasma, this is plasma, it will be B antibody. This is antibody. This is B antibody in case of A blood group people, and this is A antigen present in the surface of the red blood cell. Now, just opposite thing will be happened in the B blood group. This is the red blood cells like that, and here the B antigen will be present on the surface of the red blood cells. This is a B antigen. This is a B antigen, and here the A antibody will be present like that. A antibody will be present in the plasma. A antibody will be placed in the plasma. Now, if we coming to the, this is the A antibody, and this is the B antigen on the surface of the red blood cell. In case of AB group blood, what happened? AB group blood, both antigens will be present on the surface of the red blood cells. Both antigen will be A and B, though both are present. Both are present. Both are present. And here in plasma there will be no antibodies in case of group AB blood. And group O, what happened? No antigen on the surface of the red blood cells, but both antibody will be present here. This is B antibody and this is A antibody will be present here in the plasma. So, what happened? In this case, A antigen will be present in the group A, so that it is the self antigen of particular this blood group. So, the antibody will be B, no, not the A antibody will be present here. That's why it will be not happen. And in case of B group blood, B antigen will be present, but A antibody will be produced in the plasma. And in case of AB group blood, when both antigen will be present, there will be no, because they are both are, in case of AB blood group, they both are self antigen. So when self antigen will be produced, that antibody, that corresponding antibody will be not produced by the body, because it recognized that it is self antibody. So this is the common funda, and this A antigen is the modified form of H antigen, A and B antigen both. A antigen is produced by the some enzyme in the alpha 1,3N acetyl galactosyl amynyl transferase enzyme. And in case of B, the enzyme is alpha 1,3 galactosyl transferase enzyme. And in case of A antigen, the N-acetyl galactosamine sugar will be produced. And in case of B antigen, the D-galactose carbohydrate will be present, will be produced. So in this way, this cannot be, you have to be, when we are transfusing any blood, it should be the same blood group, otherwise O, because O has no antigen present there, and only antibody will be present there. There will be no problem, both type of antibodies are present in the O blood group, because this quantity of the transfused blood is very less as compared to the acceptor blood. Donor blood is very, the antibody will be very less, so it will be diluted in the total volume of the acceptor blood. So in this case, O group is considered as the universal donor, and AB group is considered as the this is universal blood donor, and AB group is considered the universal plasma donor. Why? Because the plasma of the AB group blood group does not contain any antibody, so it is very safe. And if you transfuse A group blood with B group blood, then there will be problem, there will be antibody, it will be the body will recognize it as a non-self antigen, and antibody will be produced, and they will be coagulated, clotting will be happen, and it may be life threatening also. So during the time of the transfusion of blood, we have to be very careful about the selection of the blood group. And O blood group can be given to anybody, but an AB blood group can accept any blood group, because they don't have any antibody in there. So this is the whole thing is based on the recognition of the immune system of non-self and self antigen. So today this is enough for today, and next video I will discuss another thing.